Hello, everyone. My name is Gert Lawler, and on behalf of the Foundation for Conservation of Atlantic Salmon, I would like to welcome you to today's webinar. Today's webinar will be presented in English. However, we have AI translation available. If you wish to use the translation, please navigate to the chat where you will find instructions on how to connect. For today's webinar, we're very pleased to be hosting Knut Vollset. Dr. Knut Vollset is a research professor at NORCE, Norwegian Research Center, and leads the Bergen Telemetry Network, coordinating research on fish movement and aquatic ecosystems. He is a member of the Norwegian Scientific Advisory Committee for Atlantic Salmon, VRL, and contributes to the National Traffic Light System, which assesses aquaculture impacts on wild salmonid populations. Dr. Vollset has authored over 100 peer-reviewed publications with research spanning salmonid ecology, telemetry, marine biology, and environmental monitoring. Today, Knut will be speaking on the perils of predicting the future, Atlantic salmon and marine survival. After this afternoon's presentation, we'll open the floor for questions and answers. And now I would like to welcome Knut and turn the webinar over to him.
Thank you, and thank you for hosting me again after that event of not being able to make the computer work last time. So my name is Knut Vollset. I'm at the Laboratory for Freshwater Ecology and Inland Fish Research Group in NORCE, and within that we have the Bergen Telemetry Network, where we do various projects on acoustic telemetry or tracking animals. I'll be talking today about the perils of predicting the future Atlantic salmon and marine survival. And for those of you that was hoping for a presentation related to predictions models to the future, I have a little bit at the end of that, but it will be a bit of a winding road till we get to that, because I will go through various things that are impacting marine survival of Atlantic salmon in Norway for the most part, which is of course relevant for European salmon as well. Just quickly, we're a group that works on various species, not only Atlantic salmon, but cod and spiny dogfish. But our main activity is related to the freshwater stage of salmon rivers, and our group has a smaller subset of people that works in the Bergen Telemetry Network. So if you're interested in animal tracking, please visit our website, Bergen Telemetry Network, and see some of the projects. But we also have a lot of focus in our research group in total on Atlantic salmon. And these days, my good colleague Helge Skogland and others are out snorkeling and surveying rivers in Norway. In the area we're lucky with very clear water rivers, and we can do population surveys by snorkeling. I'm also a member of the Scientific Council for Wild Salmon in Norway, where we are. I'm actually right now at the hotel for a 2-day meeting with this group of people, and we do assessment of all the population of Atlantic salmon in Norway every year and the risk to them. It's on the backdrop of the fact that in Norway and in Europe in general, the marine survival for Atlantic salmon has gone down. This is a time series of survival over the few data series that we have on marine survival in Norway, IMSA, showing the relatively dramatic decline in survival of 1 and 2 sea winter salmon. This reflects the general population numbers of Atlantic salmon coming back to Norway. This is an assessment of how much, or an estimate of how much salmon has come back to Norway. And the blue shaded area is the fish that was caught during sea fisheries. The brown is the river fisheries, and the green is the remaining population in the rivers. So conservation and efforts and management has led to that we are now able to actually have a larger spawning population in the rivers in Norway than we had before, but it has been due to the fact that fisheries has been severely reduced over time. In fact, the proportion of salmon, of the salmon on left to spawn in the rivers of Norway have increased from in the 80s, maybe as low as 20%, to today's numbers is a 70% proportion of salmon remaining to spawn. This is, of course, a success story, but it's mainly due to a very dramatic reduction in the sea fisheries and river fisheries, as you can see here at the same time. Our assessment of the threats to wild salmon has been done according to a risk assessment that we do every year. I'm sorry for the Norwegian in some of the slides, but I'll try to explain. What we do is that we assess the effects and the development of these risks on a XY axis. Go through a standardized form and then assess various impacts or risks to wild salmon and categorize them according to these two axes. And the color of the points also indicate how certain we are about the foundation of knowledge about these impacts. And in Norway, the three upmost highest risk effects to salmon has been identified as salmon lice, escaped farm salmon, and infections related to fish farming, together with climate change as being the highest risk factors to wild salmon. So what I'll do is I'll try to utilize the life cycle of salmon and sort of migrate through the life cycle to show some projects and some studies that we've done to assess the impacts of on wild salmon. And we'll start in the river. I won't talk a lot about the riverine stage because we're focusing on marine survival in this presentation. So we'll go straight up over to the smolt stage, where the salmon turns into smolt through smoltification and then migrates towards the ocean. In that phase, we use acoustic telemetry to study their survival and migration. But one of the issues that we've seen studying Atlantic salmon in both lakes and estuaries and fjords is that studies has shown strange patterns in some cases. And what we've discovered is that in many cases, in Western Norway at least, we see that the tagged fish that we have are actually transmitting from the bellies of predators. So to solve this problem, we focused on developing tools for identifying predation events, meaning that we haven't developed them, but we tested various types of predation sensors and tags, and studied one in particular, which was the a tilt-based predation sensor. So the tilt-based predation sensors works this way, in the sense that it gives a tilt in when it's inside the fish, and then it tilt value is according to a standard, and then if it dramatically changes, it indicates that the tilt has gone through probably the intestines of a predator, and the tilt changes, and therefore the tag starts sending out a different signal. And if you have information or have receivers close to the tag, you can also see that when it disappears out of the fish, the tilt sort of flatlines because it lays on the bottom. So with this, some of the important findings that we do is that there is a very high level of predation in lakes and estuaries of smolts, much higher than we have anticipated from standardized telemetry studies, and that in many cases sea trout can migrate similarly to smolt, which indicates that they can look like smolts but in fact are inside predators. This impacts the way that the smolts behave as well. This is a study that we did on the behavior of salmon smolts through lakes, showing that the sea trout is in the surface through most of the day, while the smolts does extensive diel vertical migrations through the day to avoid predation. And then as the season goes by, it gets more and more close to the surface because it gets desperate to get out of the lake. So the take home from these stages of study is that early migration is full of potential predators, and it's important to sort of take that into account when thinking about studying marine survival. So if we migrate further, the next element that I have focused quite extensively on during my research has been this parasite, which you can see in the zoomed in on the fin of this post-smolt. And it's this crustacean here, which is the salmon lice, and you can see this as you can see that this crustacean is a parasite because it has two legs, which is transformed into hooks that it uses to lodge onto its host when it gets close to it. The problem with parasites, and this sea lice parasite is not that it's a new parasite introduced by fish farms, but the fact that fish farms will increase the density of these parasites in the environment. So this illustration by Cecilie Nielsen shows this in the sense that the wild salmonids and sea trout is the one that first infests a new fish farm in an area, while the fish farm increases the density as a host and creates ideal condition for population growth of sea lice, and therefore it leads to a higher density of sea lice spread to the wild population. And it's a little bit important here to sort of get a bit of numbers on why this is a problem. There was a study a few years back now which estimated the proportions of parasites that are attached to farmed salmon versus wild salmon in Norwegian coastal waters, and they estimated that 97.6% of all adult female salmon lice in Norwegian coastal waters are on farmed fish. So even though farmers, the number of sea lice on each farmed fish is relatively low, because they have to keep the lice levels down to 0.5 to 0.2 female lice per farmed fish, the total number of hosts and therefore the total number of lice producing sea lice is very high because of the farm fish. And this, of course, can be illustrated by the sheer numbers of farms in the Norwegian coastal waters. They are very good at adhering to the limits set by the government to limit spread of lice by anti-parasitic treatment, but the fact that there are so many farms and so many farmed fish leads to that we have a problem with sea lice in these regions. So just to document that there is an association between the infestation pressure from farms and the number of lice on wild fish, here are some examples. There are multiple more and still coming, showing that there is a very clear, although with a lot of variance around it, a very clear association between the infestation pressure from farms and the number of lice on wild fish. Also, there's increasing numbers of laboratory studies showing that increased number of lice leads to increased probability of mortality for these post-smolts. But of course, just seeing that there are more lice on fish doesn't necessarily translate into reduced number of wild salmon returning from the ocean. So therefore, there has been various studies using what's called randomized control trials, which takes hatchery reared fish, treats half of them with antiparasitic treatment, and then releases them into the estuary of the river, and then sees the difference between the antiparasitic treated group versus the not antiparasitic treated group. And the overall meta studies we did back in 2015, now 16, showed that there is overall a higher survival of fish if you treat them with antiparasitic treatment. This was updated in 2023, where we had a longer time series, and most of the studies in and around Bergen, where we are now, has been done in the Vollset system. And the problem with the Vollset system is that very few fish comes back. So it's both been release groups, which is the R here in the actual rivers, but also release groups that's been towed out into the fjord to increase the survival of the fish, to tow them past predators, but also past farms, and then release them at various locations. So the color here indicates sort of where the 3 different release group regions. The blue is the riverine or estuarine releases, the green is the inner fjord, and the orange is the outer fjord. And then for this particular study, we also, or this particular article, we also used a station where we have counts of sea lice on wild sea trout to try to use that as a predictor of the effect of this treatment. An important aspect in these studies, which is shown to sort of be described as the golden standard of understanding the impact of sea lice, is that we've started to see some potential biases in these studies and risk of biases in these studies. For example, in this study, we found that the natural log risk of treatment, meaning that if it's higher than 0, you have a positive effect of treating them against sea lice, has declined over time. So from the start, it was very high values. This is on a natural log scale, but then over time it has decreased and are now insignificantly different from 0, indicating there is no positive effect of treatment in the later part of the time series. Of course, this could indicate that the farmers has become better at treating and therefore are better at sort of mitigating the impact on wild fish, which would be a great story to tell. However, if you look at the infestation pressure that we surveyed through surveying sea trout with these trap nets, there's the opposite trend in actual infestation pressure on wild fish. Since 2009 to 2021, where we have a time series, it's been an increase in number of lice. And you can see the number of lice here on the y-axis of salmon going from 0 to some fish having more than 300 sea lice per fish. The question then, of course, becomes if the treatment effect is decreasing and the infestation pressure is increasing, that would potentially indicate that the effect of lice is not as we expect. There is a small effect of treatment. There's a small effect of infestation pressure in this time series also on the risk ratio, particularly in the release groups in the inner fjord, which has migrated past more farms, but it's relatively weak. What we did find, however, is that if you just correlated not the difference between the 2 groups, but the actual survival in the group, you find a clear negative effect, indicating that both the treated and untreated group has a decreased survival when infestation pressure is high. This could indicate, and probably is an indication of the fact that emamectin benzoate no longer works against sea lice because the parasite has developed resistance to this treatment. A similar Irish study on this, also as from a time series a little bit earlier, has also studied the same elements and shown a clearer effect of infestation pressure. And this infestation pressure is taken from the number of lice per farm and the number of fish, estimated number of fish in the farm in total in the area where the fish has been released. And in this case, we find a positive effect of infestation pressure from farms on the risk ratio of treatment. But we also find here the same element of a negative effect in both the treated and the untreated group, indicating that sea lice treatment doesn't necessarily treat them 100% against this effect. So the conclusion from this is that there is potential biases in these types of studies. And this is just a list of potential biases that we discuss in these studies. But I would like to highlight one paper that we published a few years back, showing that we also tested by releasing pit-tagged fish in the river and then recapturing them as they were migrating out. We tested whether the emamectin benzoate treatment actually gave a lower survival in freshwater, and it did, and indicating that there might be a negative effect of these higher values of emamectin we're giving them. And the storyline here is that as emamectin has become resistant, the solution to this in some of these studies has to been to increase the dose of emamectin benzoate to still maintain the effect of it, but it has potentially shown that that actually has a negative effect on the treated group, which of course would have a reduced effect on the risk ratio between the treated and untreated control group in the survival. There is one international meta-analysis that's been led by Marie Lie Larsen on all potential studies on the effect of sea lice on marine survival that is now under review. This was quite an extensive meta-analysis and critical assessment of all potential studies, and I don't really have time to go through everything, but a very important aspect of this study was that we built a critical assessment tool to assess the risk of bias in all potential studies related to the effect of sea lice. We assess the potential of sea lice for these different criteria. We assess the risk of bias in all these potential criteria for all the studies, and the take-home message is that over time, as these studies has been done, there has been a relatively high risk of bias because of the design of the studies and way that they've been conducted, and that over time it has become better and better. But we're still, to date, when these studies are being done, the risk of bias is still relatively high in many of the studies, and that is not because necessarily the researcher are doing a bad job, but because these type of studies are really hard to do without any type of risk of bias. Also, in that meta-analysis, you could extract the risk ratio from the release group studies, and also in here we found a potential or a positive effect of treatment overall in all the studies. We also were able to extract the lice infestation pressures from the regions in Norway where there has been release group studies. And the overall main finding from that meta-analysis is that there is an effect of baseline survival on the risk ratio. So the effect of treatment is higher when survival at sea in general is poor, and also that there's a high risk of dying due to sea lice, or a higher potential positive effect of the antiparasitic drug when infestation pressure from farms are higher, which makes logical sense. The meta-analysis on overall show that the treatment increased survival on average by about 13%, which means that there will be 15%, which means that it will be a 30% fewer adult returns in the untreated salmon group, which would be wild salmon if it could be translated to wild salmon from hatchery fish. But if you can also calculate the maximum potential effect, when the maximum infestation pressure in Norway was at its highest, the reduction would be up to 62%, and when the maximum infestation pressure in Ireland was at its highest, it was a 30% reduction illustrating that effective farms can be high if they don't control the number of lice in the farms. But it's also important to point out that all studies had a high risk of bias due to methodological and reporting limitations, including poor documentation of treatment efficacy, adverse effects, and local lice pressures. And these shortcomings suggest that the true impact of sea lice on wild salmon population is likely stronger than indicated by these randomized control meta-analysis. So the next question in the marine survival aspect is, does this necessarily translate to less salmon returning in the rivers of wild fish? And the answer to that is, well, yes. In Norway, we've the same author, Marie Larsen, showed that there's a negative association of sea lice from fish farm on recreational fishing catches on Atlantic salmon. And this is particularly clear in the areas where the assessment of effects of sea lice has been the highest. So this is a map of the Norwegian coast and different production areas, and the production area 3 and 4, which is red here, is the ones that has the highest abundance, highest density of farms. There are also some non-lethal effects. We've shown in an earlier study back in 2014 that there is an increase age of maturation when fish are not treated against antiparasitic treatment, and there is a decreased size of maturation, indicating that they grow slower when they have this parasite on them. Okay, so in Norway, this study and this understanding of things has led to a management system which is called the traffic light system, and this is a system which assesses the mortality of from sea lice from farms on wild salmon using various type of tools. So they're using, for example, virtual post-smolt models. They're using trawling of post-smolt salmon to assess the number of lice on the wild fish as they migrate. They put out sentinel cages to assess whether or not there is high infestation pressure in the area. And then they use these trap nets to catch sea trout, which is in and along the coast all year round, and assess the number of lice on these fish. So this is it's just a sea trout with sea lice on them. The model then can be used to predict the infestation pressure, and the virtual population models from the different institutions in Norway. This is from Institute Marine Research, the Veterinary Institute, and SINTEF has all built their own models for sort of assessing the population effects of sea lice. The problem is that they show somewhat different patterns depending on the model assumptions. So what we've done in the recent years to be able to sort of make an assessment and an expert evaluation is that we've used what's called a elicitation method for experts, and then the different experts are asked anonymously to assess the likelihood distribution of mortality, and this is then used to create a likelihood distribution, and it's this type of information then is provided to the government to assess whether or not you should decrease or increase the number of farmed fish in an area. I have a few slides for this, but I think I'll skip this. The conclusion anyways is that salmon in Norway at least migrate through a high infestation pressure and an area of high infestation pressure and an area of high predation pressure. So it starts off kind of difficult in itself. The next question is what's going on when it migrates out from these fjords and into the ocean. And an important aspect to understand from the salmon ecology is that this is not very scientific, but it illustrates the point here, and that is that Atlantic salmon has a life history which can be compared to other. For example, herring has a growth per year that is approximately 50 grams per year. Mackerel grows from 50 to 100 grams per year. Cod grows about 200 and 300 grams per year on average. And like the Formula One fish of the ocean grows somewhere between 600 and 3.5 kilo a year, which indicates really rapid growth for this species. To compare, salmon is like in Formula One car when it comes to growth. It grows really fast, and it has a life history which indicates that it should try to get as much energy as possible in the time it's there. We have like a view into what's going on with growth at sea through the salmon scales. So salmon scales can be used to assess the the age of the fish, but also it can be used to assess the growth of the fish by looking at circuli spacing, which is the distance between the different circuli that's put into the scales, but also the distance between the smolting and the first winter at sea, for example. So then you can back calculate how fast it grew the first summer at sea until winter. One important aspect to understand in Norwegian salmon population and marine growth is that from the data series that we have from 1990 to 2004-ish, the data looked like this, where the southernmost population grew the most, partially because it was longer at sea, because it migrates earlier from the rivers. But then after 2005, the growth collapsed quite dramatically. If you correct for the days at sea, because we know when the different population migrates from the sea, you can see that actually what happened was that the four different regions from north to south grew similarly per day, but then after 2005, all the southerly population decreased dramatically. The northern population remained the same. This was also reflected in the proportion of adult fish returning as more than 3 kilos, indicating that it was a larger proportion of the fish started maturing as multiple sea winters. And the question is why that happened so dramatically. And in a study we did, we focused on the Norwegian Sea, where the salmon smolts has to migrate through. And the first indication of that there was something going on here was that we saw there was a step shift in the temperature in this region. And then to understand more why that happened, you have to go sort of under the hood of the ocean and look at the currents. And if you sort of generalize the currents, you have the Atlantic water masses moving northwards, but then you also have these two cold currents, Arctic current pumps pumping Arctic water into the Norwegian Sea. And talking to oceanographers and marine biologists, this is a really important aspect of the productivity in the Norwegian Sea, because what has happened over time is that the proportion of Arctic water masses into the Norwegian Sea, which is a little bit hard to understand from this map, but basically more red, more Arctic water into the Norwegian Sea. And then if you quantify the proportion, you get a plot like this. So over time, before 2005, what happened was that the Arctic water masses disappeared completely from the Norwegian Sea and was low for a long time. This coincided with a dramatic drop, not exactly around 2005, but various times, but in and around 2005, where the plankton, zooplankton biomass dropped by 50% in most regions. Also, the mackerel in this area, which is one of the big pelagic fish species in this system, when we measured the length of 6-year-old mackerel, the growth of these 6-year-old mackerel declined dramatically 6 years after 2005, indicating they were also impacted by the same process that were impacted that impacted salmon. So what's going on here? Well, there is some trawling of post-smolt out in the Norwegian Sea that's being caught as bycatch in mackerel and herring surveys. And this study by Kjell Utne showed that for the time data that they have, of course, they didn't have data exactly in and around 2005, but the time series before indicated the very high condition factor above 1 up to 1.2, and then after 2005, particularly in the latest year, the condition factor has been closer to 0.8, which is a quite dramatic change. Also, Ransgaard and colleagues also show that one of the things for predicting the number of fish returning is that you can use the proportion of multi-sea winter fish in the catches or in the number of fish returning, and trying to correlate that with growth or average body mass at first winter at sea. And you would expect perhaps that when growth is better, you would have a larger proportion of one sea winter, for example. But what we saw is that that relationship worked before 2005, but it changed completely after 2005. So this plot just shows that if you try to use the relationship between one sea winters and the proportion of multiple sea winters and growth to predict how much fish is returning, then you're going to fail completely if you use the same relationship before and after this, what we call, at least I've been criticized for this, but what we call a regime shift. That's one of the aspects that's going on. And of course, to understand furthermore what's going on at sea, we need to understand where the fish is migrating more specifically. And the study by Mork et al. with the modeling study showed very nicely sort of why we think that the post-smolt are in the Norwegian Sea and where they migrate. And they follow this current up and along the Norwegian coast and towards the northern parts. An element I just have to briefly mention in this context is that a new and emerging threat potentially that we haven't talked much about yet is the plans to create offshore activities. I believe personally that putting structures into pelagic areas can create habitats for fish, which is great for the fish there, but it can also potentially create habitat for predators that will potentially predate on early stages of both marine fish but also salmon. So that's something to think about. But looking at sort of where fish are being caught as post-smolts, the Norwegian salmon is probably closer to the coast than British salmon, because this is the salmon caught out here in the Norwegian Sea is probably mostly from the British Isles, and there's much more up here which follows a different current than the ones down from the British Isles. We don't know exactly where the salmon is at sea, but we have some information about what they do as kelts, which is the fish that survives from the rivers and then survives spawning and then migrates out. And there's been quite a lot of GPS tagging in and around Europe. And so this is the data which was published in 2021 by Audun Rickards and colleagues, showing that the goal of Norwegian salmon at least, but also Danish salmon, is to get up to the Arctic front and forage there. So an important aspect of not only the post-smolt growth, but also the productivity of the Arctic front, where the adult salmon adult salmon feeds, is the current situation and the productivity of the Arctic front. And up here, of course, Norway also is at Svalbard, where the the currents are shaping a lot of the ecosystem in and around Svalbard. And this is why we are also working up here at Svalbard, this is from Longyearbyen, where I was last week, and we have a PhD student that's working on, this PhD student working on tagging Atlantic salmon here, as we're able to catch them. And so if you want to know more about what's going on with the Atlantic salmon at sea, you can follow Sarah and Barre's PhD. But another aspect that can be mentioned, I think, which is important to mention in the context of this change in the Arctic, is that there are changes to which type of salmonids are going to be the climate winners. So this below here is what we think will be the climate loser, which is the Arctic char, and the above here is what we think will be the climate winner, which is the new invasive pink salmon that we've started to get into Norwegian rivers. Pink salmon is a risk to Atlantic salmon. We think we don't know very much about the potential impact, but just the storyline behind the Atlantic salmon or pink salmon invasion is that it was Russia that first introduced them in the 70s and 60s. Didn't manage to introduce them. They wanted to establish a fisheries, didn't manage to introduce them then, but in the 80s they tried to introduce it again from a river called Ula in the Pacific, and they managed to introduce it. But the number has been low over a long time. But then in 2027, it suddenly, probably because of climate change and the same processes that has led to this increase in growth in the north, has also led to a recruitment of pink salmon. So this is just a plot of the number of pink salmon over years. And of course, if you don't know, pink salmon recruits and dies, and has 2-year cycles where the returns are in Norway every odd year. So 17, 19, 23 and 25 is a pink salmon year. And of course, they are definitely eating all the same things as Atlantic salmon, but also other things because they're smaller as they go out. It is interesting to understand what's going to happen with this pink salmon population. The potential for its establishment is huge, not only here in Norway, but also in places like Greenland, and probably, give it time, it will also come to Canada on the east coast there. Just to give you some numbers, in 2023, more than 460,000 pink salmon was caught or observed in Norwegian rivers, and that compares to the total number of Atlantic salmon that came back to Norway rivers to spawn, which was 323. So our salmonid, most abundant salmonid in Norway now is pink salmon. Trying to sort of migrate back into the rivers, another aspect that we have problems with in Norway is farmed salmon. It is a threat to genetic diversity, and also, I would say, a threat to the general view on the farming industry and Norway. This story of farmed Norwegian salmon escaping in Iceland rivers was a big problem, both for Norwegian industry, but also at Iceland. We've shown that the number of farmed fish in rivers are tightly linked to the abundance of farmed fish in the fjords, which sort of makes sense, but also gives a clear management indication of what to do, I guess. Finally, we also study things that's going on as they migrate back. And one of the interesting aspects there is that we've been able to reestablish otter populations in Western Norway in areas where where wild salmon are doing very poorly. And by tagging and following Atlantic salmon, we've established how much in some of these populations is being eaten by otters. And this is a master student and Robert Lennox from OTN tracking salmon and that potentially been eaten by otters. In these rivers, these relatively small rivers, we've estimated in some cases that otters can eat 95% of the remaining spawners in rivers in years where marine survival is really poor, and provides a really tricky environmental dilemma that we have to consider. And maybe we'll have to choose between otters and salmon in some of these rivers in Western Norway. Okay, so I'm almost done, but I thought I'd mention predictions, because I said predicting the future, and one of the assessments that things that we've been asked to do is to make prediction models for the future because of last year's horrible salmon year and the fact that they closed quite a lot of rivers. So government in Norway said that we have to make prediction models. So I'll try to run very quickly through some of the things that's been going on there, and that is that we have data from all the rivers of Norway, and we can assess whether or not we can see whether or not the time series of Atlantic salmon can be used to predict next year. So I'll go quickly through this, but the model that we built so far is an assessment using generalized additive models to assess 1 sea winters, and then 2 sea winters, and then 3 sea winters as a functions of the environment, and the 1 sea winter the year before and the 2 sea winters the year before. The environmental drivers that has been shown in the prediction model to potentially explain some of the variation and numbers of fish coming back predictably into the futures is herring biomass, the inflow of Arctic water, the temperature in the Arctic, and the estimated mortality from sea lice. So it sort of follows some of the studies that I've shown so far. So what we do is that we use a training set from time series in the beginning, and then we predict the next year, and then we continue like that to assess whether or not we're able to use these data sets to predict next year's number of salmon. Yeah, anyways, this is just an output from just the sort of the GAM model to show you sort of what lays behind here. And this is very simply sort of the correlation between the total number returning and the total number predicted returning, which kind of shows that it sort of works. We're maybe able to predict some of the numbers, but in some rivers we're definitely not. Like down here on the right hand side, there is definitely poor predictions. And if you start looking at the actual predictions and the numbers, you start realizing that they are quite poor for various types of rivers and classes of rivers. Sorry, but if you look sort of overall on average across these, we are able to give some predictions with quite a lot of uncertainty around us. The black are our predictions, and the red are the actual numbers returning. Of course, what we should be able to do is assess very poor years, and in some cases, and the model per date doesn't necessarily do that, but at least we have some grasps around what's going on. So that was my final slide. I was a little bit over time, but thank you for listening. And if you're interested in any of the work we do, please contact me on this email.
Thank you so much. That was an excellent presentation. We will now open the floor for questions. If you have a question to ask, please click the hand icon to raise your hand. The moderator will see this hand and call on you to unmute. That would be me, and ask your question. Please double check that access is granted for the GoToWebinar program to access your microphone. You may be prompted with a pop-up to allow access. Click allow and then unmute to ask your question. This would be the preference, but if this function does not work, you can type in your question in the chat and I will read it out loud. So I'm just going to check for questions. Okay, we have Frederick Risky. on here, and I'm just going to enable him. Okay, Fred, go ahead with your question.
Hello, Knut. How are you?
Very good. How are you?
With regards to your herring biomass predictor, is this a positive or negative relationship? Is it actually a food source for the salmon at some points, or is it a competitor? And how do you tease out those two effects within the modeling?
You can not. It's a very good question, and there is the role of using a prediction model is would just assess whether or not it works to predict next year, and sort of the way that we've sort of focused on this is that we accept the fact that we don't necessarily don't understand all the mechanism. But of course, we need to assess whether or not the fits to the predictors makes any sense. Like if they're completely off to our general understanding, we have to reassess them. But we allow sort of the GAM model to fit relatively like unmechanistic patterns. But that being said, there seems to be maybe going on both, because we have one model for 1 sea winters, one model for 2 sea winters, and one model for 3 sea winters. And the effect of herring seems to be different for 1 sea winters and 2 sea winters, which might make sense in the sense that is a negative effect for 1 sea winters and a positive effect for 2 sea winters. And that could be competition at early stages, or it could be that they choose to mature later as 2 sea winters, and it changes their maturation scheme. So we don't know exactly if it's the one or the other, but there's also a theory that it's related to it's more related to that the herring attracts predators, large schools of predators, and that may also forage on the salmon. But these are hypotheses that we can't really tease apart. So, but it's an interesting question. I agree.
Okay. Now we have a question from Dave Dunn. Dave, I'm just going to unmute you. Go ahead.
I think you can hear me now.
Yes, we can hear you now.
Okay, thanks. Look, great. It's a great presentation. Thank you very much. I kind of missed your predictive slides at the end, which I think are the crux of the presentation. I'm wondering if you could sort of go over the last slide or the last couple of slides and summarize them, and then also in the context from a North American perspective. How do we apply this information looking more towards the migratory route towards Greenland? Is it relevant, and what portions of it are relevant or are not relevant? Would you be able to answer that at all?
My knowledge on sort of the eastern coast Atlantic salmon, the ocean currents there are fairly limited, unfortunately. But I think, I mean, the general understanding is sort of the ocean currents and the productivity of for post-smolts are probably very important. I think I've seen studies on this saying that what they think when it comes to growth is that European salmon are more bottom-up driven, productivity driven, and where there has been like historically a growth relation between the growth and survival, while in eastern Canada, and this was when I collaborated with Kevin Friedland, they indicated that there might be more a top-down predator effect on sort of the very low survival there. I don't know if that's the case, but I would sort of definitely sort of think about and reconsider if you can try to understand sort of where where you potentially could measure the productivity and the recruitment of prey for post-smolt is for these post-smolts. When it comes to trying to go through again the way that this is done, This is fairly preliminary in the sense that we're still working on this, and probably going to continue working on this. And I'm presenting one. So I'm presenting one method, and we also have 2 other methods that we're working on that I could, when we do write the report, that will probably be in Norwegian, but when we do finish the report, I can definitely give you more details. But on one side is a regional assessment, which uses just regional number coming back, and my sort of task, and that makes it much easier to use regional data to identify potential predictors. And then I've taken these predictors and put them into individual river assessments and tried to sort of tease out more details on the potential impact using this modeling approach. And then in addition to that, there is machine learning methodology, which we're going to apply afterwards to see. Now we're doing modeling exercises where we, as a person, as a modeler, has to choose the different sort of combination of things and exact what sort of combination to test at the best, and then try to test this predictive power. But using the machine learning, you can sort of make the machine learning do all that process faster, and then potentially get prediction models which are even better. So I don't know if that describes it better in the process, but if you're interested.
Yeah, thanks for that. Yeah, it describes it a little bit better, and I think it was unfair to perhaps ask you to comment on the Eastern North America seaboard. But anyway, it was a good answer, and thank you.
Give it another minute to see if anybody else has questions. Okay, that seems to be the end of the questions so far. If you do have more questions later, please feel free to reach out to Knut directly using the email that he has provided on the screen, or contact us at the foundation and we will certainly pass it along for you. We've recorded the session and it will be available on our YouTube page. If you just search under FCAS-FCSA, you will come up with our channel. Our next webinar session will be held on Wednesday, November 12th. Andre St-Hilaire will be presenting on the topic of recent advances in temperature modeling in Atlantic salmon rivers. So big thank you again to Knut and to everyone who participated today. We hope to see you at the next webinar.